This is our first study, pilot study funded by Global Food System to look into the antimicrobial properties of sorghum phenolic compound, which is an antibiotic alternative on very important production disease of cattle, that's liver abscess. So, as many of you know, the beef cattle industry contributes close to 8.2 billion to Kansas economy, accounting for close to 18% direct contribution to our agriculture sector. And liver abscess is an important production disease of cattle, the beef cattle. And the primary liver abnormalities of feed cattle. The problem with the liver abscess is until the animal goes to the slaughter, you never know that animal has a liver abscess. That is the problem. So again, the main contributing factor or a predisposing factor for liver abscess is the high grain feeding during the finishing stage of close to 40-50 days, sometimes close to 120 days. It depends on the feed lot. And again, the liver abscess is very common in Holstein cattle compared to crossbred cattle. So the prevalence ranges from close to 10 to 20%. Again, it varies from breed, type of diet, and the geographic region, and other various various factors. So again, the liver abscess is mainly caused by bacterial pathogens. The primary pathogen of liver abscess is Fusobacterium necrophorum and Fusobacterium funduliforme, which is always present 100% in almost all the liver abscesses. Again, the size of liver abscess, as you can see, might vary from a golf ball to a football size. So then the secondary pathogen is Tuberella, and recently, back in 2015 and 16, we isolated Salmonella enterica for the first time from liver abscess. So till now, Tylosin, which is an important macrolide antibiotic, is approved by FDA for control of liver abscess in feedlot cattle. But as you know, the future of Tylosin is uncertain because of the development of antimicrobial resistance, because most of the bacteria, gut bacteria, are getting resistance to Tylosin. And since the January 2017, the FDA has come out with a veterinary feed directive, so where the feedlot producers have to get a consent or a veterinary oversight or a veterinary feed directive from a veterinarian to feed Tylosin in the feed cattle. So that led to the considerable interest in evaluating antibiotic alternatives. So one such alternative is phenolics, phytophenol, any plant-based phenolic compounds, and probiotics, essential oils, and medium-chain fatty acids. These are commonly tested in cattle feed. So the phytophenols are known to have antimicrobial properties because they are antioxidant in nature and also anti-inflammatory. But till now, most of the phytophenols, they don't know the mechanism of action, but they say that they are antimicrobial. So we wanted to test the hypothesis. And then again, because they contain the plant-based extracts, most of the plant-based extracts are contain a lot of phenolic compounds. Thus, the hypothesis is they might have a potential, they might act as a potential natural antibiotic alternatives. So our objective for this proposal, for the pilot study, was to investigate the efficacy of sorghum phenolic compounds on the liver abscess-causing bacterial pathogen. Again, we procured black sorghum, sumac sorghum, burgundy, and white sorghum. Dr. Yang Li played a pivotal role in getting all those stuff, and then his lab did a lot of major chunk of work. Again, this was done in Dr. Yang Li's lab. Again, I am representing both my collaborators. So the phytophenols were extracted using 75% aqueous acetone. And again, the total phenolic content was, or TPC, was determined by spectrophotometer. And this table shows the TPCs, the total phenolic content for sumac sorghum, black sorghum, and burgundy sorghum. And I didn't include white sorghum because the white sorghum had a very negligible content of TPC. So then what we did was we tested the phenolic compounds by using two different approaches in the lab. So we did a macro dilution method, which is qualitative, where I will show you the pictures. So we tested these phenolic compounds in different concentrations, and we measured optical density, which is an indirect measurement of bacterial growth. And we also did a viable bacterial cell count, just to make sure that it's really working. Then we also we wanted to quantify, now that it worked, we wanted to quantify at what concentration this phytophenol can kill bacteria or inhibit the growth of bacteria. So that was a quantitative assay. Then all our analysis was done using SAS. We used a ProgGLM procedure to fit a linear model, and analysis of variance was performed on log transformed MIC value. And model included fixed effects of tested phenolic extracts and replication as a random effect. And all the assays were done in replicates and duplicates. So again, my students are working here. So macro dilution method, the bacteria were cultured in a medium with and without phytophenols. And again, we at 6 hours, 12 hours, and 24 and 48 hours at hours intervals after incubation, the bacterial concentrations were determined by optical density, and also viable cell count were estimated by spread plating method. So as you can see, this is one of the, this picture shows the effect of sumac sorghum phenolic extract against Truparella pyogenes. As you can see, this one, the left panel has a control where you can see a lot of bacteria. At 24 hours, sumac sorghum was able to kill all the bacteria. In 48 hours, nothing, So, which was statistically significant. Again, the black sorghum, we had the same result. And burgundy sorghum, we didn't find any effect. So, then we wanted to quantify whether really it's working. So, this is the result of microbial dilution. As you can see, this table, all the tested sorghum phenolic compounds, they didn't inhibit Fusobacterium and Salmonella bacterial species, but Truparella had an MIC of 42.5, 23.7. Again, these are in microgram per ml. And this is how these are the microtiter plates where we quantify the inhibitory effects of phytophenol against bacteria. To summarize, the phytophenols from sumac and black sorghum inhibited Truparella pyogenes, and the sumac sorghum phenolic compound had more inhibitory effect, as you can see, because sumac sorghum had more TPC, total phenolic content, which is directly correlated to their effect. And the phytophenol that inhibits the pathogen may have the potential to control liver abscess. And again, further studies are ongoing. Because of COVID, we were unable to, me and Angie Lee, we planned a trip to Texas to get some varieties of different genetic lines of sorghum. It didn't happen. So Dr. Varaprasad is helping us to get some different genetic lines from Mexico. So black sorghum varieties. So again, the sorghum work started. So we used part of the fund to do this kind of work also. Like one of my students was finishing a PhD, so we wanted to test this in collaboration with USDA. This is how all the sorghum work started before I collaborated with Dr. Lee. So we also used we used some part of the money from like a global food system to test foodborne and mastitis causing pathogens. Again, mastitis is another economically important production disease of cattle. And the foodborne illnesses are zoonotic, and most of them are of public health concerns. And we tested 3 major bacterial foodborne pathogens, such as Shiga toxin producing E. coli, Salmonella enterica, and Campylobacter. And again, CDC recognizes these species as one health importance. And mastitis again is an important disease. Common bacterial pathogens of mastitis in dairy cattle are Staphylococcus aureus, Pseudomonas, and Klebsiella. Again, because of the antimicrobial resistance, using antibiotic is not permitted. So that's why we thought that we can use a phytophenol. So this table shows that the black sorghum varieties we got from USDA back in before submitting proposal to GFS was found to be effective against Campylobacter, Staphylococcus aureus, and Enterococcus faecalis. So again, we also found, just like our studies, we found an upward dose-dependent trend for antimicrobial activity. So increased dose of phytophenol, so increased activity against bacteria. Again, our study provides impetus to test additional cultivars, which we are planning to do right now, of sorghum, and explore value-added benefits of sorghum grains. So right now in the lab, we are planning to do simulate the same thing what we did in the lab in rumen of cattle. So we have some 9 rumen calculated animals. We're trying to dump all the sorghum stuff as it is, like how we feed the cattle, like cattle feed. Then we are testing the concentration of liver abscess-causing pathogen. Again, these are the products from this proposal. As you can see, the first 6 my collaborators, my mentor, Dr. T. Nagaraja, is the main person on liver abscess with 9 different patents on liver abscess. So the highlighted ones, my DVM student, the award-winning abstracts in a national meetings or proceedings. And the last one is my PhD student worked with in collaboration with USDA. Again, we also disseminated the findings in our Beef Cattle Institute podcast, so which is called Cattle Chat, which is very popular. So we do 2 times a week, 2 times a month actually. so which was telecasted in podcast. And we also have a peer review publication in Antibiotics Journal. So again, the GFOS was acknowledged. Because the major funding for this project was given by Center for Sorghum Improvement Program, SARA. And again, with this, I would like to acknowledge the Global Food System and then Cancer Sorghum Grain Commission and VRSP for funding my student. So we have a veterinary research scholar program funded by NIH. And my collaborators in USDA and SARA section, those are again, Dr. Lee is here, is an integral part of our proposal. So I'll be happy to take your questions.